Variation is inherent to Western blotting, and the sources of variation, both biological and technical, need to be identified, understood, and managed. There are many sources of biological variation. Here are a few examples. If you work with cell lines, you may already know that the confluency of cultured cells has a major impact on protein expression profiles. Confluent cells, such as epithelial cells, are engaged in plasma membrane remodeling and formation of adherent junctions, while subconfluent cells are committed to log phase growth. Environmental stimuli, such as temperature and light exposure, can also cause profound effects. Temperature changes are well known for affecting enzyme reactions, even to the point of denaturing the enzyme. And anyone who has raised chickens or horses is aware of the effects of lengthening light exposure during the seasonal change from winter to spring. 14 to 16 hours of light will drive the hormonal changes that occur for reproduction in many mammalian and avian species. Variation during developmental stages is ubiquitous. One interesting example is the relative changes reported in transcription expression levels for the termite salivary cellulase. Expression of this transcript is highest in the worker termites and nymphs, which is considered consistent with the feeding habits of these forms. Expression is very low in the pre-soldier and soldier termite stages, which are the forms that defend the colony. Technical variation in Western blotting can be introduced a variety of ways, including unequal total protein sample concentration loaded on the gel, pipetting error that can include incomplete aspiration or dispensing of volume and drift of sample into other wells, edge effects on gels such as smiling, frowning, and distortion of end lane samples, poor gel to membrane transfer efficiency that can be due to protein size, transfer buffer makeup, transfer apparatus, transfer voltage and time, as well as heat generation, and exceeding membrane binding capacity. Managing variability means knowing where it can creep in and then carefully designing and executing your experiments to reduce error by keeping timing, temperature, protein concentrations, etc., as consistent as possible. Reducing or eliminating error before applying a normalization method will improve sample reproducibility and, importantly, minimize the contribution of normalization to data analysis. Always keep in mind that normalization is a strategy and not just a step in your protocol.